let's look at a type of inheritance that's called incomplete dominance. So it's a good name because the dominance, the phenotype dominance, the appearance is incomplete. So with incomplete dominance, the F1 hybrids, meaning that F1 generation, have an appearance, a physical appearance, their phenotype, that's in between the phenotypes of the parents. So in the example here, here's the P parental generation, a red flower and a white flower. They're crossed, and you have a F1 generation that's a hybrid and happens to be heterozygote. So this is similar to what we first saw with the Mendel's peas, where maybe you had a yellow pea crossed with a green pea, okay? And then the offspring was the dominant color, right? Not in between. But with incomplete dominance, you get an in-between phenotype, right? If this was dominant recessive inheritance, like in Mendel's peas, this individual should be red, because notice that R, for the red color allele, is dominant, capital R, and then white is the recessive phenotype for the recessive allele. But the heterozygote here is not red, it's not even white, it's in between. If you mix red and white, you get pink. So how is this explained? It's explained via incomplete dominance. What happens is when the individual is heterozygote like this, not all of the dominant phenotype is expressed. So some of the recessive phenotype is seen. So it's a blending, it's a mixing of the two phenotypes in the homozygote of the homozygote dominant and the homozygote recessive. So the F1 generation, the heterozygote, since it's in between in terms of its phenotype, that's explained through what's called incomplete dominance. Oh, sorry. Okay. And then if you do F1 cross, notice that the gametes can be containing a capital or lowercase allele, right? A dominant or recessive allele. And then notice the F2 generations. There's their genotypes, but notice the phenotypes. You get a red, white, and two pinks, because the two heterozygotes express a phenotype that's in between the homozygous recessive and the homozygote dominant and recessive. Okay? All right. So incomplete dominance, and it's seen, you'll be able to know if it's incomplete dominance by examining, understanding the phenotype of the heterozygote. So something called hypercholesterolemia is a human condition of incomplete dominance characterized by dangerously high levels of cholesterol in the blood because of the inheritance pattern. Heterozygotes have intermediate blood cholesterol levels. Notice if you're homozygous dominant, you have an ability to make what are called LDL, a type of cholesterol receptors. So the phenotype is that you have normal amounts of cholesterol in your blood. Okay? You can think of this as the healthy, normal condition. On the other end, if you're homozygote recessive, you do not make the LDL cholesterol receptors, and you have severe high levels of cholesterol in your blood, and you're severely diseased. You'll have some serious cardiovascular problems as you age. However, if you're the heterozygote, notice what's happening. It's a heterozygote condition. It's not the dominant condition or the recessive condition. It's in between. So you have a mild case of the disease. So again, since the heterozygote is in between the homozygous dominant and the homozygous recessive conditions, this is explained by incomplete dominance. Right? The dominant phenotype is not fully expressed, incomplete dominance. Again, it's the heterozygote that allows you to determine what type of inheritance pattern we see. All right. So, excuse me, other types of inheritance. Multiple alleles that lead to what's called codominance. Codominance. So not only one allele, but more than one allele gets expressed in codominance. So classic example is the human blood types, our ABO blood groups. There are actually 3 alleles in the human population: the A allele, the B allele, and the O allele. And the O allele means that the A allele surface molecule on the blood cell and the B allele surface molecule on our red blood cells is absent. So in the human population, there are 3 blood type alleles, A, B, and O, in the simplest case. There's actually quite a bit of others, but we'll keep this as simple as possible. So the combination of the 3 alleles produce 4 common blood types. Okay? Now, we all have 2 alleles because we inherit them from our parents. So if you're A, right? Homozygous A, you're going to be blood type A. If you're homozygous A and absence, like you don't have a second allele, you're still blood type A. If you are homozygous BB, you're blood type B. If you're B, we can call it 0 or absent, you're blood type B. If you have the alleles A and B, then you're blood type AB. In other words, your red blood cells have both the A and B red blood cell surface molecules present. However, if you're blood type O, that means you do not have the A and you do not have the B allele. So phenotypically, you do not produce any red blood cell surface molecules. So then you are blood type O, right? So in the human population, we're either blood type A with those 2 genotypes. Blood type A is the phenotype, or your blood type B with either of those 2 genotypes, or your blood type AB with that genotype, or your blood type O with that phenotype, the absence of the A and B alleles. Now you've probably heard of blood type like O negative or positive. We're not going to consider the positive or negative because that's yet another allele. Okay, so the 4 blood groups that can consist, as I talked about, blood type A, ability to make substance A, which is a red blood cell surface protein molecule. Okay. That one for B, which is here, or lowercase I with no allele for neither A or B, and that will give you blood type O. So here you go. Notice if you're blood type A, it's because you have the presence of the A allele and you produce phenotypically the surface molecule A. Blood type A. If you're blood type B, that's because you have the allele B and you produce the red blood cell surface molecule, which we'll just call B, so you're blood type B. But if you're blood type AB, you can think of this is the heterozygote because it's not the same, right? It's one and the other one. That's the heterozygote genotype for blood type A and B. What do you produce? Not only A or not only B surface molecules, but A and B. You were expressing both. Both alleles are dominant in the sense that they are both expressed. So this is called codominance. Right? So people with genotype A and B make both surface molecules, carbohydrates. Sorry, down here, right? Notice both of them. So both alleles are expressed in the heterozygote individuals, so they have blood type AB. And again, the way to distinguish this type of inheritance pattern is by examining the heterozygote. The heterozygote, again, will tell you if it's codominance or incomplete dominance. All right? So things can get even more complicated. With pleiotropy, and the example of sickle cell anemia or sickle cell disease. With pleiotropy, there's more than one gene or allele. Well, the correct way to say this is with pleiotropy, the gene or the allele will influence more than one characteristic. Okay? If we look at this, the allele A determines the molecule A. If it's allele B, it determines the molecule B. Right? So one allele determines one phenotypic characteristic. All right? A and B, well, A and B, right? The A allele, A molecule. The B allele, B molecule. Each allele determines one characteristic. With pleiotropy, each gene or allele will influence several characteristics, more than one. So with sickle cell disease or sickle cell anemia, the abnormal hemoglobin proteins that are produced by the sickle cell allele, so there's the gene or the allele, it produces a malformed protein, sickle cell hemoglobin. Now that malformed protein will misshapen the red blood cells and give them this sickle shape, kind of a crescent shape. That's why the disease is called sickle cell anemia or sickle cell disease. And here's the red blood cells that are sickled. Why? Because of the malformed hemoglobin molecule, which is used to carry oxygen. So the one allele or gene produces at least two characteristics here. Misshapen hemoglobin molecules, which are too small to see, but that influences the misshapen entire red blood cell, which we actually can see on the slide. Okay? Then the misshapen blood cells can clog small blood vessels called capillaries, and it can lead to pain, organ damage, and even paralysis. So it's a whole cascade of events from simply one malformed or dysfunctional gene or allele. So this is called pleiotropy. So with genetics, things can get somewhat complicated. We're trying to keep them as simple as possible. So if you're homozygous recessive, these individuals have the sickle cell allele, both of them, and will have a severe case of sickle cell disease. So it is a recessive condition, sickle cell disease, but since the defective gene or allele causes a whole cascade of problems, physical manifestations, this is called pleiotropy, okay? With one gene or allele influencing multiple characteristics. Okay, now polygenic inheritance. Polygenic, look at the word. Many, polygenic, meaning alleles or genes influencing some characteristic. So this is how we explain why people are expressing a range of heights, right? Not everyone is 5'2" or 6'2", right? But people are expressed by a whole continuum range of heights, okay? Or weights, you know how much we weigh, or hair color. There's a whole gradation from very, very light hair to very, very dark hair. Eye color, skin color, right? When you plot out a characteristic, phenotypic characteristic, and you have enough people, you'll get this normal bell curve distribution. Very tall individuals, a few of them. Very short individuals, a few of them. Most individuals are right in the average height. How is this explained? It's not dominance, codominance. It's not recessiveness, dominant inheritance. This is explained by polygenic inheritance, where multiple genes or alleles have an additive effect on a single physical phenotypic characteristic like height. Okay. So height in people is controlled by several genes that are inherited separately on different chromosomes, and how those genes get expressed will determine how tall we are or how short we are. Okay. And the same for skin color, eye color, hair color, the texture of our hair, many, many characteristics, even to a certain degree intelligence. Okay. Polygenic inheritance, multiple genes, alleles influencing some phenotypic characteristic. Right? The phenotypic characteristic is the physical characteristic of individuals or organisms. Okay? So now epigenetics and the role in the environment. We do not grow up in some sterile bubble. We grow up in the environment, and the environment has an influence on how our genes are expressed, and sometimes a significant influence. And this is what epigenetics talks about, and what I will talk about in the next section.